My name is Sean Hervey Jumper. I'm a neurosurgeon here at University of California, San Francisco. It has been just such a privilege to work with patients and researchers and all the staff here. And my interests have really lied at the intersection of cognition and brain cancer, both as a clinician and as a cancer neuroscience researcher. I think it's essential to begin with a broader overview of how the adult brain maintains normal function and supports cognition and cognitive operations in the non-disease setting, just in the normal setting as we live and age. So cognition, cognitive processing requires both cellular level, meaning from how cells interact, from both neurons, these are the cells that transmit a signal from one to the other, and neurons to glia. Glia are the supporting cells that help the neurons do their job better. And these connections, while collectively establish this network or the circuits, and these circuits in the brain is really what underlies or what sets the framework for cognition and understanding of how the brain actually functions and communicates from one part of the brain to the other. Throughout our lives, there is some recovery that happens. We call it adaptive neuroplasticity. And that's a process of maintaining healthy cells and their dynamics, predominantly through the hippocampus and a population of cells that are called neural progenitor cells, or NPCs. These cells promote the hippocampus and other regions of the brain to make new cells. Another term you'll hear is oligodendrocyte precursor cells, or OPCs. These are the types of cells that maintain the important structural elements of the brain. They make the connections stronger, and they wrap connections with something called myelin. It's these processes that are strongly influenced by the microenvironment of the brain, and they are the underlying mechanism that helps cognition and helps you maintain normal, healthy brain function. So the brain's ability to reorganize itself by forming new neural connections throughout life is neuroplasticity. So neuroplasticity allows the neurons or the nerve cells in the brain to compensate for injury or for disease and to adjust their activity in response to specific situations or changes in the environment. And intrinsic brain tumors such as glioma is one change in that environment. So again, I have to bring one important point up in the context of this talk, which is that the definition of neuroplasticity in the biomedical science world is much more narrow than we'll use it today. So neuroplasticity, when you read a paper, really involves a functional change that's measured at the protein level or structural level in the brain. We're going to talk a little more liberally about neuroplasticity. The more lay definition of neuroplasticity is any functional recovery or functional change that happens over time. So we're taking a slightly more liberal context to this plasticity term